This is a production of Cornell University.
All right, so the students out there, when you are always practicing your seminars, because it's for moments like this when you have to do performance under pressure, when about 6 of your slides got chewed up by the PC or my flash drive or something. So I'm going to do my best to fill in those gaps, but hopefully it won't be too serious. All right, well, the first thing I want to do is I'll thank the people who did the work towards the end, but the people who funded the work, long-standing funding to my lab from the Department of Energy, both to my lab and then through this EFRC that we have at Purdue called C3 Bio. Some postdoctoral support for a fellow by the name of Nick Bonowitz from the LSRF, and we've also had funding from GCEP, a funding agency through Stanford, NSF, and our collaborator John Ralph in the GLBRC. So what I want to talk to you about today is what I think is just testimony to how fortunate I am that we did a mutant screen many years ago. I discussed the first of the mutants that came out of that screen when I interviewed at Boyce Thompson in 1993. I got the job at Purdue, though, which is where I met my wife, so that's okay. Everything worked out just fine. But it's a mutant screen that we started, and it took us down the road of enzymes, of phenylpropanoid and lignin biosynthesis, but more recently, with this screen, we identified an important regulatory component, a part of the mediator complex, which is why I sort of have this oblique pun in here about mediate, that turns out to, the function of which turns out to explain a number of the unexpected phenotypes we saw in our biochemical mutants. So, it's going to be a little bit weaving and crossing over that I hope I can walk you through all this. So first of all, why are we interested in studying phenylpropanoid metabolism? I've had a long interest in secondary metabolism, why plants make all the fabulous compounds that they make. But phenylpropanoid metabolism is one that's particularly important both biologically and in terms of the use of biomass for humans, for bioenergy or animal feed, or even for paper production. But for plants, it was one of the important innovations that permitted the colonization of land, because it involves the generation of molecules that can absorb UV light that the plants now have to deal with if they're on land compared to living in an aquatic environment. So this happens to be the molecule that's going to feature prominently in this talk. It's the UV protective molecule that Arabidopsis makes, called Sinapoylmalate. And it's, of course, this portion of the molecule on the right that is responsible for absorbing UV. And the reason that I show this portion of the name in blue is that the great feature about this for us is that this moiety makes this molecule fluorescent under UV light. So as a mutant phenotype, the presence or absence of this fluorescence has been very useful to us. The other important component that is derived from this pathway is lignin. And this is sort of a schematic representation of what lignin might look like. We still don't have a very good understanding of the actual structural. Well, we know the structural motifs, but not exactly how it exists in the wall. And of course, this molecule, I like to describe it as being important at both the macroscopic and microscopic level. So that the lignin polymer is impregnated into the cell wall, where it gives it rigidity and allows these trees to stand upright and withstand the forces of wind and what have you. So it's a very important at that level. But at the microscopic level, it's also important as a molecule that strengthens the cell walls of the water conducting tissue, the xylem. So here's a cross section of a Arabidopsis stem, and you can see the tracheary elements that are nice and big and open and good for conducting water. And in this mutant that has lower levels of lignin, those same cells have collapsed under the tension, of course, that's developed by transpiration. Now, I don't want to leave you with the impression that lignin's the only important strengthening agent. A cellulose deficient mutant will show a similar phenotype. So it's clearly a codependent network. So anyhow, it's pretty clear that this pathway is very important. Now, many of you probably aren't lignin experts, but have heard a lot about it with regard to biomass and biofuel production. Why is that? Well, one of the reasons I would argue is that it represents low-hanging fruit. It's a unique polymer in that, well, let's go through the other ones that we're all familiar with in plants or in organisms. Nucleic acids, we know that their synthesis is template dependent. Protein synthesis, we're reading an mRNA template to make a protein molecule. Polysaccharides, well, that's a little bit different, but still there are specific enzymes that make specific linkages to make very characteristic polysaccharides in a cell wall. Lignin synthesis is entirely different in that it's the types of crosslinks that you see in the lignin polymer and the components that make it up are dependent only upon the precursor supply. So whatever so-called monolignals that a plant makes, whatever ratio the plant makes them in is represented in what the polymer is like. Now, let's just contrast that. If you had a yeast cell that overproduced leucine, you would never expect to see its proteins containing more leucine. But here we have found, and I'll show you some of the data, well, not really data, pictures, that we have that show that we can manipulate monolignal synthesis and thereby manipulate lignin, both chemistry and, it turns out, architecture. So lignin monolignal biosynthesis begins in the cytoplasm with the amino acid phenylalanine, and there's a series of hydroxylation and methylation reactions that lead to these three alcohols: paracumaryl alcohol, coniferyl alcohol, and Sinapoyl alcohol, so-called H, G, and S subunits for lignin. And so you can see that what we're talking about is differences in the decoration of the ring. So this is the earliest branch off the pathway, the second one, and the third one. And I'll show this slightly differently in some upcoming slides. So these molecules can then be exported to the apoplast, where they're activated to free radicals by lacases and peroxidases, which then spontaneously polymerize to form the polymer. So depending upon what monolignals are made, that determines what comes out and what is polymerized. So here's a representation of the pathway that probably is sufficient for most people's purposes. I assure you I know the structures involved, but if I showed them all, you wouldn't be able to see them and you wouldn't remember them anyway. So I don't know, maybe it's teaching 20 years of undergraduate biochemistry that's driven me to this. But so this is my schematic of the pathway, and it again starts with phenylalanine and branches to give the G subunit coniferyl alcohol or the S subunit Sinapoyl alcohol. I want to show this because this molecule I mentioned earlier is a derivative of this same pathway. So Sinapoyl malate comes off of the pathway fairly late. So by looking for mutants that are deficient or reduced in Sinapoyl malate accumulation, we have been able to identify enzymes, genes and enzymes that are not only required for these final transformations, but also a number of the steps that are common to both Sinapoyl malate biosynthesis and lignin biosynthesis. And so as I go through my talk, I'll often use the accumulation of Sinapoyl malate basically as a proxy for overall activity of this pathway. So as I mentioned, Sinapoyl malate is fluorescent blue under UV light, and you can actually see that in planta. And so here is a wild-type plant photograph just with a transilluminator for illumination. And the first mutant we identified that we called FA1 for ferulic acid hydroxylase it completely lacks Sinapoyl malate, and you can see it has this nice red fluorescence that comes about because, well, first, there's no blue fluorescence from Sinapoyl malate, but also because Sinapoyl malate is an effective UV sunscreen, in its absence, that UV can penetrate down into the mesophyll, excite chlorophyll molecules, and you see chlorophyll fluorescence. So if we were to take this photograph again with a yellow filter over the lens that would block out this blue fluorescence, what you would see here is a red plant and a plant that's basically just dark, because this is an effective sunscreen. It blocks UV from getting into the mesophyll. So what I'm going to talk to you about today is a number of these mutants, the later ones of which we refer to as REF, for reduced epidermal fluorescence. All right, so I mentioned that we can manipulate lignin biosynthesis, and I mentioned that partially in terms of the justification for the research, but also it's important for what's going to come later in my talk. So this is very old work, so I'm just going to mention it very briefly. So if we eliminate F5H, ferulate 5-hydroxylase, in the Fa1 mutant, this was the first time that we had shown that we could do this lignin manipulation. So in the absence of that enzyme, the lignin is derived entirely from G subunits. And the effect on the plant is essentially there is no effect. These plants grow and develop normally, and that's perhaps not surprising because if you look at outside, pine trees, gymnosperms make only this G lignin. So we already know that's an effective polymer in some plants. And so these Arabidopsis plants are fine with that. And in fact, we've also found that if you overexpress this step, we can redirect flux over to make a pure S lignin. And again, it has no effect on the plants. It's a functional lignin. It's quite interesting because it's easier to degrade. And it actually has, instead of being a cross-linked polymer, it's a linear polymer. So we're really dramatically changing the cell wall structure, but it's tolerated very well. Now, that's not always the case. So we have an example of a mutant that is very early in the pathway, C3'H. And when we block that step, the plants make a lignin that is derived from these H subunits. These subunits make up normally about 1% of the polymer in a wild-type plant. But the lignin in these plants is almost entirely derived from that, but that has very severe consequences. So that's what that plant looks like. And so we were not maybe entirely surprised by this. In the lignin literature, it was said that you couldn't even make H lignin. And so we thought, well, this is probably not a functional polymer for some reason or another. So the fact that these plants are dwarf, perhaps it's got to do with, and I'll just use the same slide as I used earlier, this idea that the abundance of lignin is important. Those plants make very little H lignin, and also show this vascular collapse. And we know, obviously, that water transport is very important to plants. So a plant that can't transport water is dwarf. Not a terribly big surprise.
Are the proportion of the different lignin cell types specific?
Yes, in many cases. So the xylem has predominantly G lignin, and that's both in Arabidopsis and also in tree species. Whereas supporting tissues, fibers, sclerified cells, deposit S lignin. But usually, especially in the case of those cells, it's when I say S lignin, it's really a GS copolymer. So does that answer your question? Yep. So I'm going to set that mutant down for a moment and talk about another of the mutants that we've worked on called REF4. This turns out to be the regulatory protein that I mentioned at the beginning of my talk. Although when we first identified it, we didn't understand that to be the case. So REF4 mutants also deposit less lignin than wild type, so you're getting used to seeing these. Now you can see that in the strong allele, we have collapsed xylem compared to these cells, and we have about a 25% reduction in overall lignin content. And that's highly consistent with the literature. That's about the extent to which you can reduce lignin before you start seeing phenotypes like this. These plants are also dwarf. You can see here on the right, so this is the strong allele. But once, of course, we had cloned the gene, we could identify knockouts for this to see whether they would recapitulate this mutant phenotype. And this is a knockout for REF4, and you can see that it does not. This is actually a missense mutation that is semi-dominant. So it wasn't really a huge surprise that the knockout didn't recapitulate that phenotype. REF4 has a paralog in the Arabidopsis genome that we call REF4-related 1 or RFR1. A knockout of this also doesn't phenocopy REF4-3, and the double knockout also does not. So there's certainly something special about this particular mutant allele with this. It's a very minor change in the protein structure. It's a glycine to serine alteration. But what it appears is that that mutation causes that protein to act as a constitutive brake on phenylpropanoid metabolism. So we were very relieved, in fact, to find that the knockouts, or the double knockout particularly, gives the opposite phenotype. So here's wild type. This is using, again, sinapoylmalate levels as a measure of phenylpropanoid activity. You knock out REF4, there's really no change. You knock out RFR1, you do get an increase, and the double knockout has an even more substantial increase. So the fact that this is a great relief, because the fact that this gives the opposite phenotype to this suggests that this is not a neomorphic mutation. So this is actually a protein that's acting in the same pathway it normally acts in. So again, for the students, if you've not thought about this before, if you think about a neomorphic mutation, what about a transcription factor that has a missense mutation that leads to a change in the amino acids on the DNA binding domain that now allow it to bind somewhere where it wouldn't normally bind? Now you start studying that, now you're studying a context for that transcription factor that's not biologically relevant. So here, the knockout having the opposite phenotype to this dominant mutation was a great relief to us. Okay, so what is REF4? Well, we didn't know for the longest time. It's found in all plants. It has no domains that allow you to, it has no ATPase domain. It doesn't even have a domain of unknown function. And so we were actually left scratching our head for the longest time about what this thing is really doing, until somebody purified the mediator complex from Arabidopsis and REF4 immunoprecipitated with the rest of the complex. So what is mediator? It's a large 30 protein transcriptional regulatory complex that can associate with another kinase module and bring in yet another 4 proteins. And what it does is that it's divided into a number of modules, and this so-called tail module interacts with transcription factors bound to DNA, both locally and at a distance. And when it does so, the head and middle module can recruit to the transcription start site the general transcriptional machinery. And so in doing so, by making contacts with a number of transcription factors bound upstream of genes, it can generate it can sort of interpret that information and lead to a coordinated transcriptional output. So what we had identified was MED5, a subunit of the tail. This is really fairly fanciful, but if you look at the EM reconstructions of it, they're a little bit hard to interpret. So in some ways, the schematic is more useful. But anyway, MED5 is the protein that we've been looking at. So we now have, we understand now that we've got a protein that's involved in phenylpropanoid regulation. We have a missense allele that suppresses phenylpropanoid gene expression. Now, for reasons that are, well, entirely serendipitous, in my lab, we were making multiple mutants. My postdoc was, and we can now circle back to this REF8 mutant that I mentioned earlier that was blocked earlier in the pathway and made only H-lignin. Nick made a triple mutant, in fact, between REF8, this very sad plant, and this knockout of these two flavors of this Med5 mediator subunit, and found that these plants were capable of growing again, which was a big surprise, because we thought, well, this is all just to do with malformed lignin and lack of ability to transport water, but these plants grow just fine. So that was quite a puzzle to us, but it was very exciting. What Nick found was that was actually able to restore lignin deposition. So again, this is a measure of lignin content. You can see in the dwarf mutant, it's very much suppressed, but when we bring in the mediator mutations, that lignin content goes back up. So maybe it is the fact that the case that these plants can transport water properly. They certainly look like they can. But also it's not the case that we had in any way restored normal phenylpropanoid metabolism. These plants are still blocked at the same place we thought that they were blocked before. And where we could really see that clearly was by doing the lignin analysis. And this is work done in John Ralph's lab by Yuki Tobimatsu. And if you were like really interested in lignin, you'd look at this and go, wow, but you're not. So let me explain why you should be excited about it. So this is 2D NMR and lignin analysis, and the blue stands for the G subunits, the green for the S subunits, and these red resonances here are for these H subunits that are normally just very, very minor components. And so these plants that are growing normally are making this type of lignin that people thought plants couldn't make. So that we thought was pretty exciting. So now we've actually, what I've introduced along the way are plants that make GS copolymer, that's the wild type, pure G, pure S, and now pure H lignin. So you can see how this polymer can be very readily manipulated. Anyway, for those of us in the lignin community, that was pretty exciting. So why is this mediator mutation restoring growth? Well, the bottom line is that we still don't know, but I can tell you a couple of things that aren't going on. First of all, it's not the case that the low lignin is because phenylpropanoid gene expression for lignin synthesis is downregulated in the plant, because in fact, in the REF8 mutant, it's upregulated. All of these genes required for lignin synthesis are upregulated in the mutant, and they're further upregulated in when we knock out the mediator subunit. So it's not that we're restoring overall gene expression. It's certainly something more complicated than that. To try and get a handle on that, we did an RNAseq experiment. And what you can see is that, well, there's a number of things I guess I should point out. So on the left are the upregulated genes, on the right are the downregulated genes, and we're comparing the mediator subunit double knockout, the lignin mutant, and then the triple mutant from both. So there's a few things. One is there's a huge number of genes that are misregulated in that very dwarf plant, and that's probably to be expected in a plant that is so developmentally perturbed. There are relatively few genes that are altered in expression when you knock out this mediator subunit, which we're continuing to study this in the lab. What is the impact of altering mediator on gene expression? And in many cases, it's surprisingly modest for an important, what we've always considered to be an important component of this transcriptional regulatory apparatus. And in the triple mutant, almost all of the genes, over 90% of the genes that are misregulated in the mutant are returned to normal. Okay, so before I go to that, so again, we still haven't identified the actual cause from all of these. There's too big a number of genes to be able to focus in on what the actual cause of that dwarfing is. But we're almost inclined at this point to say that it's not that the low lignin causes the dwarfing, but perhaps it's the other way around, that we're actually altering metabolism in such a way that we get this massive transcriptional response that is mediator dependent. So that's a sort of a theme that I'll carry through the rest of the talk, that perhaps in these plants, some metabolite is being detected that accumulates because of the block in lignin biosynthesis, and that leads to mediator-dependent changes in gene expression. Okay. So now I'm going to go on to my next sort of case about, or a little vignette about mediator and how it may function in regulating metabolism, both within pathways and between pathways. So this is not an experiment, it's an anecdote. It comes from when I was a postdoc. So in the early 1990s, before some of you were born probably. And it was when I originally identified the FAL1 mutant. So this is the one that makes the pure G lignin. And what you can see here is two pots of two different alleles of FAL1 and wild type. I had been growing these plants in a nice growth chamber, and I needed the space for something else, so I moved the plants to some place, I don't remember, I think it was higher light and a bit warmer temperature. And the wild type plants did what wild type plants are prone to do, and they were upset with being moved, and so they accumulated a lot of anthocyanins, commonly thought of as being stress metabolites. And yet the FAL1 mutants did not do that. And I was really perplexed about that for about 25 years. And we decided to try and follow up on that a little bit. You can maybe people in the front row can see, many of you who have worked with Arabidopsis know that Arabidopsis petioles and stems, there's a little bit of anthocyanin there. That's missing in these FAL1 plants. To try and get a clearer signal, we use this line called PAP1D, which is an activation tagged line that turns on expression of a transcription factor, which is a master regulator of anthocyanin production. And you can see this very dark red pigmentation in the petioles. Just it drives up the levels that are where they are normally found. But when you cross that into a FAL1 background, that doesn't happen. You don't get that level of anthocyanin accumulation. And this was really perplexing to me. Oh, yes, I can show you this again. In cross sections of stems where you, again, get a lot of anthocyanin accumulation in PAP1D, but in a FAL1 PAP1D double, you fail to get anthocyanin accumulation in all the cells that would normally express that gene. So it seems that it's clear that there's some interaction here, but the perplexing thing about this is that this enzyme of phenylpropanoid metabolism is not required for the production of anthocyanins. The anthocyanins branch off very early from the pathway, and sinapoylmalate and S-lignin very late, and yet we're seeing this interaction between the accumulation of these products. And that was very perplexing. And we could see that if we did HPLC analysis of those anthocyanins. In wild type, there's a large peak or a couple of large peaks of anthocyanins when we measure by HPLC. The red line represents the levels of anthocyanins in the FAL1 mutant. It's very, very low. But when we compare MED5, the MED5 mutant, we see anthocyanins just like in wild type. But when we combine MED5AB and FAL1, we restore anthocyanin accumulation. In this case, it's a little bit complicated. It's a different anthocyanin that accumulates because this anthocyanin actually has a synaptic acid subunit on it. So that decoration is missing, and so we get a different anthocyanin here. So when I say F5H isn't required for anthocyanin accumulation. I mean the core of the anthocyanins, not the decoration. So this effect of Fa1 on that of suppression of anthocyanin accumulation is mediator dependent. What we think is happening, and I'll show some models a little bit later, is again, perhaps a metabolite upstream of F5H is being sensed in a mediator-dependent way and is shutting down phenylpropanoid metabolism. Okay, this is, I think, where, yes, this is where I go into the part of the talk where my slides got corrupted. So I'll do my best. The next mutant I want to tell you about is this mutant called REF2, and another one that isn't shown in this slide, REF5. For those of you who were here when my postdoc interviewed Dr. Jung-Im Kim, this is the story that she told, so perhaps you'll be able to fill in the gaps that are missing in my slides. So these two mutants, REF2 and REF5, are low in Sinapoylmalate, as that UV phenotype reveals, and we can show that by HPLC as well. So again, Sinapoylmalate content, and in these multiple REF2 alleles, it's suppressed. We've shown a long time ago, this was from 2003, that REF2 encodes a cytochrome P450 dependent monooxygenase known as CYP83A1. Now the confusing thing at the time was that these P450 monooxygenases are hydroxylases by and large. But all the hydroxylases of the phenylpropanoid pathway have been accounted for by that time. It turns out that this P450 functions in a different pathway. Wait a minute, let me go back. Let's watch this letter here. REF5 encodes the paralog of REF2, and that's CYP83B1. I'm sorry, I'm missing data off this slide, so I can't show you the Sinapoylmalate levels. But this is work that Jung published in 2015. Now, this is where I have to do like interpretive dance or something instead of showing you the pathways. But both of these enzymes function in glucosinolate metabolism. Most of you are probably familiar with glucosinolates, yes? But for those of you who have not, at least you have, if you like wasabi, for example, because it's the horseradish, or mustard, it's those glucosinolate degradation products that give those products their flavor. So CYP83A1 and CYP83B1 are enzymes of glucosinolate biosynthesis, and yet a distinct pathway from phenylpropanoid metabolism. Distinct, you can see on the right, it's distinct from the pathway on the left. It isn't there anymore. And yet these blockages at these enzymes are affecting phenylpropanoid metabolism. What Jung-Im went on to show was that, so this is a more in-depth analysis of this REF5 mutant. What she showed was that if she blocked in the REF5 mutant, CYP83B1, Sinapoylmalate levels were downregulated. If she blocked the enzyme before it in the pathway, Sinapoylmalate levels were actually elevated. And this genetic lesion is actually epistatic to this one. So if you make a double mutant, these Sinapoylmalate levels are elevated. So that says two things. One is it looks like it could be a compound between this block and this enzyme that's responsible for leading to the downregulation of phenylpropanoid metabolism, and also that this crosstalk between glucosinolate synthesis and phenylpropanoid metabolism is relevant in a wild type context, because when you knock it out, Sinapoylmalate levels actually go up. So here is a little bit of a schematic that did survive the slide debacle here this morning. And so the idea here that is that there are two types of glucosinolates that are made in Arabidopsis: methionine-derived ones, in which the CYP83A1 functions to form an aldoxime as an intermediate, and tryptophan-derived glucosinolates in which CYP83B1 functions, again to form aldoximes. This crosstalk appears to somehow affect phenylpropanoid metabolism, and Jung Im, by doing a suppressor screen, so she took the REF5 mutants, remutagenized those, and looked for plants that were blue in the M2 generation. She found, in a thing which a discovery which I still feel like we're extremely lucky, turns out to be, the suppressors turn out to be Med5, one of the two subunits. So this crosstalk, again, now between two pathways rather than within one pathway, is dependent upon mediator. And she also went on to show that this same suppression was seen when she crossed the Med5 mutant into the REF2 background, which is the CYP83A1 involved in methionine-derived aldoxime synthesis. So that other blockage in the previous slide that I mentioned, this one here, CYP79B2 and B3, is the enzyme that converts these amino acids to their aldoximes. So again, we block here, we get an accumulation of aldoxime, it's perceived by Med5, and that somehow impinges on phenylpropanoid metabolism. So to try and determine how that perturbation was enforced, what was responsible, what was involved in that crosstalk, Jung Im did a RNAseq experiment. And it's somewhat complicated because she's comparing 4 different lines. So she's comparing REF5, REF2, and then REF5 with a mediator knockout and REF2 with a mediator knockout. So what she was looking for, rather than explaining it through here, I'd rather use these sort of cartoons. What she was looking for to find genes involved in this crosstalk was genes that were upregulated in both REF5 and REF2, whose upregulation was mediator dependent, so that when we looked at REF5 Med5 and REF2 Med5, they went down, or the other way around. Genes that were downregulated in these mutants that went back up to normal in the double. And so those are represented by 53 genes and 18 genes, which is a manageable list. And from that, she was able to identify 2 genes that were of particular interest that met those criteria. And these are Kelch repeat F-box proteins, number 39 and number 50. So these are upregulated in the glucosinolate mutants, and that upregulation is abolished when we knock out Med5. Same over here. And what do these things do? These are proteins, or components of the process that targets proteins for degradation by the proteasome. And in fact, these KFB proteins are known to interact with PAL and target it for degradation. PAL is the first enzyme of the phenylpropanoid pathway, and I've represented the proteasome with a garbage disposal, and I apologize if there's anyone here who works on the proteasome, and I've compared your work to that. Anyway, so, and in fact, when she goes and measures PAL activity in these various lines, she does indeed see that in the lines that are upregulated for the KFB proteins, PAL activity is down, and when we bring in the mediator mutations, that the PAL activity is no longer reduced. And in fact, she's gone on to knock out those KFB proteins and to summarize a large array of different mutants. If she takes the REF5 mutant and which has reduced levels of Sinapoylmalate, as of course I've been saying, and knocks out these 3 KFB proteins, we get restoration of phenylpropanoid metabolism. So it seems that out of that RNA-seq experiment, she was actually able to identify the at least one protein that was, or two proteins that were responsible for this crosstalk. Okay, well, I want to conclude with a final story that goes back to the original mutant that we identified that led us into this mediator story. Because we thought it would be a valuable reagent to start, I guess, dissecting mediator function in plants. So mediator is very well studied in humans and in yeast, and there are a number of mutants of mediator that show its relevance to a variety of processes, flowering time and whatnot, plant pathogen interactions in plants, but we certainly are just starting to scrape the surface. So a former graduate student of mine did another suppressor screen as a way to start probing this. And so here's Columbia wild type and the REF4 mutant, the dwarf, and the three knockouts. And so my student did a suppressor screen looking, remutagenizing REF4 plants and looking for plants that were now blue under UV or big or both. And the idea behind it is to identify other proteins that are required for this dominant form of this MED5 subunit to exert its action. And so my student identified this array of suppressor mutants and handed this project off to another grad student. And what she did was quantify Sinapoylmalate levels as well as lignin, but I'll just focus on this for today, but also sequence all of those suppressors and identify the genes in which they were defective. So although each mutant carries about 200 EMS-inducible mutations in the open reading frames of the genome, by taking a candidate gene approach and looking for genes that are mutated in common, basically an in silico allelism test, we were easily able to identify the genes that were responsible. So in these mutants, we find a sort of a spread of restoration of phenylpropanoid metabolism. The black lines represent intragenic mutations in MED5. So if we've turned this protein with this one G to S mutation into a constitutive, a dominant break on the pathway, if we then knock it out, we would expect to see a reversal of that phenotype, and indeed we do, although it's at various levels. But the other thing that came out of the screen was mutants in 3 different mediator subunits, different ones. So this is MED2, MED16, and MED23. These are all in the tail of mediator, are thought to be, with MED5. And so we're right now in the well, you can see it's listed as submitted here. We're understanding how these different mediator knockouts might affect gene expression on their own and how they affect gene expression in the context of this REF4 mutation. What I think is quite interesting is that, for example, these two, I'll focus on this one, this suppressor restores, well, both of them actually, I'll take that back. Both of these suppressors, one in MED2, one in intragenic mutation, restore growth, but not phenylpropanoid metabolism. So we can pull these apart again. It's not the case that if we just muck around with mediator, we will abolish the function of REF4, because here's an example of 2 different mediator knockouts, PFT1, MED25, and MED3. And when we cross those into the REF4 background, we don't get restoration of growth. So again, it's at these these results are specific. It's not just a case of perturbing the tail of mediator that blocks REF4-3 function. Whitney went on to do an RNAseq experiment and found then, I guess, a continuing theme in that in REF4-3, one of the same KFB proteins involved in PAL degradation is dramatically upregulated, and in her suppressors, they are restored to near normal, suggesting that this PAL degradation is again, in this context, responsible for the phenotypes that we see in this mutant. So I can just wrap up now and say, well, so here are our models then. So looking at the glucosinolate example, we have a crosstalk between two pathways that is mediator dependent, where if we block glucosinolate synthesis and that leads to the accumulation of these aldoximes, these are perceived somehow by Med5. We lead to upregulation of these KFB proteins, which target PAL for degradation, and therefore we reduce the downstream components of this pathway. In the case of anthocyanins, we think there's a similar thing going on where when we block F5H, there's a buildup of an upstream metabolite that is perceived by Med5, or directly or indirectly. That also turns on KFB, decreases PAL, and in a cell, probably in a cell type specific way, knocks down anthocyanin synthesis. With REF8, it's a little bit more complicated. We do know that KFB gene expression is enhanced in this, that very dwarf mutant that we had in C3'H, perhaps again because of a buildup of some metabolite. But there's another huge set of changes in gene expression that lead to dwarfing that are Med5 dependent. So it's a more complicated story. And in the case of REF4-3, we have, in fact, a mutant form of this Med5 protein that has the ability to constitutively turn on this KFB protein, leading to PAL degradation and an impact on phenylpropanoid metabolism. So, I hope I've convinced you that Mediator is involved in phenylpropanoid homeostasis. We would desperately like to know what it's perceiving and how it's perceiving it, and we have efforts going on in the lab to try and figure that out. In that first example, the dwarfing that we've seen in low lignin plants can be suppressed genetically. We always thought it was sort of a mechanical problem to do with water transport, but it's much more complicated than that, involving mediator and aberrations in gene expression. This KFB mediated degradation of PAL seems to be a common mechanism for regulating this pathway. Previous to that, and I really shouldn't end this talk without mentioning that this was this whole idea was first identified by C.J. Liu at Brookhaven, and without that knowledge that he contributed to the literature, we still wouldn't understand what was going on. This seems so. Before that, we really thought that it was feedback from cinnamate, the product of the enzyme. So this is a really important new paradigm, I think. And I would also say that, since I mentioned it in passing, we have these H lignin plants, we've got G lignin plants, we've got S lignin plants. These plants with modified lignins are, I think, could be valuable for the biorefinery industry if that ever really gets going, because it's taking an important or an abundant product in the plant cell wall and turning it into something with more defined characteristics, and thereby could be more easily converted to downstream products of value. And with that, I guess I would like to thank the people who did the work. Nick Anderson and Nick Bonowitz. Nick Bonowitz worked on all the REF8 suppression stuff. Nick and Nick together on the anthocyanin project. Jung Im on the glucosinolate project. and Whitney and Jake on the REF4 suppressor project. And also my collaborators, John did and Yuki, who did the NMR, and CJ, and Brian Dilks for help with the sequencing and suppressor analysis. And with that, I will say thank you very much for your attention and the invitation.
Thanks very much, Clint. Any questions?
Yeah.
So I have a question about the dwarfism in the biosynthesis. So it might be slightly different angle from this matter, but there's a mutant called the Lignin mutants, which spontaneously overexpress salicylic acid, and that a lot of the Lignin mutants have dwarfed. And somehow I just think there is a link between these two because salicylic acid is also from the phenylpropanoid biosynthesis.
Right.
So is there any, do you think, like feedback mechanism if you overexpress the salicylic acid, does that suppress the lignin biosynthesis? Because a lot of people simply argue that if you have a lot of defense mechanisms, for example, like overexpression of salicylic acid, then the energy distribution of the plant would be more biased to the defense mechanism, and the growth is suppressed. So I think lignin might be the one cause of the suppression of the growth. So is that possible? Do you think?
Okay, so there's a lot rolled up into that question. So let me tell you what little I know about it. So first of all, yes, there's growth defense trade-offs. Quantitatively, lignin is vastly more abundant than SA. So whatever we're doing with lignin, although it could add to SA, I don't think how much SA a plant makes is really closely tied to lignin abundance. I would also say that one of my other grad students will be very happy to hear that you asked this question, because I didn't have time to talk about it today, but once Whitney had basically completed her REF4 suppressor analysis, my other student Candy asked whether the CDK8 kinase module, which is known to have repressive effects on transcription, could be involved in the phenotypes that we see in the REF4 mutant. So she showed that it was, that knocking out CDK8 reverses dwarfism, but has no effect on phenylpropanoid metabolism. And looking at her transcriptome data, she saw that SA synthesis, conjugation, and response genes were all upregulated. So she said, "Oh, let's test the hypothesis that SA is involved in the dwarfing," and so she knocked it out with by crossing in a SID2 mutation, and found that SID2 does not, it reduces SA accumulation as expected, but it had no effect on growth of this mutant. So I can't say it doesn't contribute at all, but there are other factors that are more important in that particular case, I guess. Yeah.
I find it interesting that the mediator is a target for regulation for like responsive since doing so many different things.
Mm-hmm.
Are there like a whole set of genes that's regulating? Are they seem to be related pathways, or things that could be like related? Like if you are decreasing the glucosinolate, you want to be changing the lignin, but it's just like it's just regulating all sorts of things.
Well, certainly there's many different pathways that are perturbed when you knock out different mediator subunits. I mean, GO analysis will always tell you that, right? No matter what set of genes you look at, practically. But with regard to about the glucosinolate crosstalk, I'm still of two minds about that whole story, and we've been trying to actually ask the question: is this as you sort of framed it, I don't think I did, but I think it's reasonable that you did, that you would want to have crosstalk to coordinate the two pathways. You know, there might be some situation where you want to use phenylpropanoids for defense, and other times when you want to use glucosinolates for defense. Okay, that's a good model. Or is it a kind of a pharmacological effect? So we know we can do a chemical biology experiment, and we can add compounds exogenously to yeast or seedlings or whatever, and see an effect on a pathway, on growth, something like that. So is this crosstalk something that operates like that, just so happens that these aldoximes have this effect? Or is this crosstalk truly important for biology? And I would think that if such a pharmacological effect exists, it must exist sort of as a precursor to the evolution of important coordination between pathways. If you know it, there's got to be something for evolution to work on. So until there is something like that, I don't know how you evolve, pardon the sort of teleology, such a crosstalk. So I'm reluctant to say that this is really metabolic coordination, although it could be. Does that answer? I'm not sure that answers your question a little bit. Yeah.
So along those regards, could you find evidence for that if there was cell type specific variation? So if you're seeing, for example, around vascular tissue, there's lots and lots of lignin being deposited, compared with, say, epidermal cells where maybe you want to have more of a UV filter. You see cell type specialization within an organ, and you see different sets of interactions or degrees of expression mediated. Perhaps that would talk to this idea that it's being co-opted to predominantly balance metabolites in different situations.
Right. So, but at the same time, we have to ask whether that's coming about because of, as a result of what Mediator is doing, or whether it's coming about because of where, just by happenstance, there's an overlap between glucosinolate and phenylpropanoid metabolism.
So, do you see any predominant expression within a leaf, for example, in certain cell types?
Of what?
Mediated?
We don't have that information. It's not, we don't see it at very high levels. And it's actually been a big pain to do the biochemistry. You have to actually be looking at, to go back to cell cultures. Meristematic cells seem to be where it's expressed most highly. But we're actually trying to get at this question by installing the aldoxime synthesizing enzymes, the CYP79, in non-glucosinolate producing plants to ask, do you see the same impact on phenylpropanoid metabolism? Because that would argue that it is a pharmacological effect, perhaps. So, there.
Question: do you know if there's any circadian regulation of F5 or the mediator complex?
I don't know. If you really see very little interesting things when you look at expression analysis of the mediator complex, and maybe that's because, you know, it's required for most regulated gene expression, and so it has to be everywhere. Again, you do see it, some of the subunits up in meristems, and whether that reflects particular role for those subunits or just higher requirements for transcription in those cells, I don't know.
Okay, well, if no further questions, please join me.
This has been a production of Cornell University, on the web at cornell.edu.